Welcome to Biology, the scientific study of life. Comparative studies help explain how the evolution of development leads to morphological diversity. Biologists in the field of evolutionary developmental biology, or Evo-Devo, as it is often called, compare developmental processes of different multicellular organisms. Their aim is to understand how developmental processes have evolved and how changes in this process can modify existing organismal features or lead to new ones. With the advent of molecular techniques and the recent flood of genomic information, we are beginning to realize that the genomes of related species with strikingly different forms may have only minor differences in gene sequences or regulation. Discovering the molecular bases underlying these differences in turn helps us understand how the myriad of diverse forms that cohabit this planet have arisen. This information, the study of evolution, widespread conservation of developmental genes among animals. Molecular analysis of the homeotic genes in Drosophila has shown that they all include an 180 nucleotide sequence called a homeobox, which specifies in 60 amino acid homeodomain in the protein. An identical or very similar nucleotide sequence has been discovered in the homeotic genes of many invertebrates and vertebrates. In fact, the vertebrate genes homologous to the homeotic genes of fruit flies have even kept their chromosomal arrangements. Notice that homeotic genes in animals are often called Hox genes. Furthermore, related sequences have been found in regulatory genes of much more distantly related eukaryotes, including plants and yeasts, and even in prokaryotes. From these similarities, we can deduce that the homeobox DNA sequences evolved very early in the history of life and was sufficiently valuable to our organisms to have been conserved in animals and plants virtually unchanged for hundreds of millions of years. Not all homeobox containing genes are homeotic genes. That is, some do not directly control the identity of body parts. However, most of these genes in animals at least are associated with development, suggesting their ancient and fundamental importance in that process. In the Drosophila, for example, homeoboxes are present not only in the homeotic genes but also in the egg polarity gene bicoid, in several of the segmentation genes, and is the master regulatory gene for eye development. Researchers have found that the homeobox encoded homeodomains domain is the part of a protein that binds to DNA with the protein functions as a transcriptional regulator. However, the shape of the homeodomain allows it to bind to any DNA segment. By itself, it cannot select a specific sequence. Rather, more variable domains in a homeodomain containing protein determine which genes the protein regulates. Interactions of these later domains with still other transcription factors helps a homeodomain containing protein recognize specific enhancers in the DNA proteins. With homeodomains probably regulate development by coordinating the transcription of batteries of developmental genes, switching them on or off in embryos of Drosophila and other animal species. Different combinations of hemiobox genes are active in different parts of the embryo. This selective expression of regulatory genes varying over time and space is central to pattern formation. Developmental biologists have found that in addition to a homeotic genes, many other genes involved in development are highly conservative from species to species. These include the extraordinary similarity among particular developmental genes in different animal species. Raises the question, how can the same genes be involved in the development of animals whose forms are so very different from each other? Current studies are suggesting likely answers to this question. In some cases, small changes in regulatory sequences of particle gene can lead to major changes in body form. For example, the differing patterns of expression of the Hox genes along the body axis in insects and crustaceans can explain the different number of leg-bearing segments among these segmented animals. Notice this figure. In other cases, similar genes direct different developmental processes in different organisms, resulting in different body shapes. Several Hox genes, for instance, are expressed in the embryonic and larval stages of the sea urchin, a non-segmented animal that has a body plan quite different from those of insects and mice. Sea urchin adults make the pink ocean shells and may have seen on the beach. They are among the organisms long used in classical embryological studies. Sequencing of the Arabidopsis genome has revealed that plants do have some hemiobox containing genes. However, these apparently do not function as master regulatory switches as do the hemiobox containing homeotic genes in animals. Other genes appear to carry out basic process of pattern formation in plants. Comparison of animal and plant development. The last common ancestor of plants and animals was probably a single cell microbe that lived hundreds of millions of years ago. So the process of development must have evolved independently in the two lineages of organisms. Plant evolved with rigid cell walls that make the movement of the cells and tissue layer virtually impossible, ruling out the morphogenetic movements of cells and tissues that are important in animals. Instead, morphogenesis in plants um, relies more heavily on differing planes of cell division and on selective cell enlargement. But despite the differences between plants and animals, there are some basic similarities in the actual mechanisms of development, legacies of their shared cellular origins. In both the plants and animals, development relies on a cascade of transcriptional regulators turning on or turning off genes in a finally turned series of, for example, setting up the head to tail axis in Drosophila and establishing the organ identities in a radial pattern in the Arabidopsis flower. But the genes that direct this process differ considerably in plants and animals. While quite a few of the master regulatory switches in Drosophila are hemiobox containing Hox genes, those in Arabidopsis belong to a completely different family of genes called the Matchbox genes, and all the homeobox containing genes can be found in plants and midbox genes in animals. In neither case do they perform the same major roles in development that they do in the other group. In this final chapter, the genetics unit, That to effect of differences in Hox gene expression during development in crustaceans and insects. Notice that changes in the expression patterns of four Hox genes have occurred over evolutionary time. These changes account in parts for the different body plans. The bring notice also the brine. Notice here, the brine shrimp, a crustacean here, this one, a top, and the grasshopper, an insect. Shown here are a region of the adult body color coded for expression of the Hox genes that determine formation of particular body parts during embryonic development. Notice that thorax abdomen, notice that, and thorax genetic segments, and notice also. Okay, back to conservation of homitic genes in a fruit fly and a mouse. Homitic genes are that control the form of anterior and posterior structures of the body, occur in the same linear sequence in a chromosome, Drosophila and mice. Each colored band on the chromosome shown here represents a homitic gene. In fruit flies, all homitic genes are found on one chromosome. The mouse and other mammals have the same similar sets of genes on 4 chromosomes. The color code indicates the parts, notice that, of the embryo in which these genes are expressed and the adult body regions that results, notice that. All these genes are essentially identical in flies and mice, except for those represented by black bands, which are less similar to the two animals. Okay, thanks.